Hi guys, we are talking about microbiology and we are Cristina, Arad, Alejandro and I, Aranza González Ruiz. Microbiology is the scientific study of these microorganisms. Microorganisms are those organisms that are too small to see with the naked eye and included things like bacteria, fungi and viruses. Microbiologists study these organisms using tools like microscopies, genetics, and culturing. Microscopies allow scientists to magnify microbial cells that are otherwise too small to see. Genetics and molecular biology help scientists understand the evolutionary relationships between microbes and their habitats. Culturing is a term that you describe growing microbes, usually combined with tests to see what the microbes like to eat or what conditions they can live in. If you are even seeing a petri dish, you have seen a common place where microbes are cultivated. Most of the microbes or bacteria in your body are called residents. These bacteria are well-established residents of your body, especially the skin and the gut. They are your first line of defense against potentially dangerous transient bacteria, and you might pick up from touching a door handle of being near someone who sins. The resident bacteria can usually outcompete the transient bacteria, preventing them from stalling in a causing infection. Infection from pathogenic bacteria can sometimes clean up of their own, or with help from antibiotics. Antibiotics are the virus medicines that fight bacteria by damaging proteins that cells wall or carrying out their damaging attacks on bacteria. A bad side to antibiotics is that they can rarely tell the difference between good and bad bacteria. With antibiotics, both resident and trans bacteria are diminished, and while it will help up your infection, it might also give you a bad stomachache. Viruses are a different story. Viruses are even smaller than bacteria and are a special classification of life. Viruses can only reproduce by using a host cells. Sometimes these can be other bacteria and sometimes they can be cells on your body. They are very simple agents, and because they cannot produce independently, they are sometimes not even considered the true life forms. Bacteria, singular bacterium, is a microscopic single-cell unicellular life form that exists practically everywhere in the Earth's environment and is simpler than other cells of animals, fungi, and plants. Of about 3 million species of bacteria believed to exist, only about 4,000 are known and are divided into 5 general groups according to their shape: Bacilli, broad-shaped; Cocci, ball-shaped; Filamentous, thread-like; Spirilla or spirochetes, spiral-shaped; and Vibrio, comma-shaped. Some are free-living, harmless, or beneficial, even essential to the other life forms. Others are parasitic and produce toxins that cause disease or death.
Hello, my name is Alejandro and I'm going to talk about of the most important systems for the identification of microorganisms. The most important system for the identification of microorganisms is observed for growth on artificial foodstuffs prepared in the laboratory. The food material in which microorganisms grow is the culture medium, and the growth of the microorganisms is the culture. We have prepared more than 10,000 different culture media. For bacteria to grow properly in artificial culture mediums must meet a number of conditions including temperature, humidity, and pressure of oxygen, adequate and proper acidity or alkalinity, A culture medium must contain the nutrients and growth factors required and must be free from uncontaminating microorganisms. Most pathogenic bacteria require complex nutrients similar in composition to body fluids of the human body. Therefore, the base of many culture media is an infusion of meat extracts and peptone to which other ingredients are added. Agar is a solidifying element employed for the preparation of culture media. It is completely liquefied at the temperature of boiling water as the solidifies upon cooling to 40 degrees. With minor exception, it has no effect on the growth of bacteria and is not attacked by thus growing on it. Gelatin is another solidifying agent but is used much less since many bacteria cause liquefaction. In different culture media are numerous enrichment materials such as carbohydrates, serum, whole blood, bile, etc. Carbohydrates are added for two main reasons: to increase the nutritional value of the medium and fermentation reactions to detect microorganisms that help to identify them. Serum and whole blood are added to promote the growth of the less resistant microorganisms. Dyes that act as indicators to detect, for example, the formation of acid or growth inhibitors of some bacteria and not others. Phenol red used as an indicator because it is red in basic and yellow pH in acid pH are also added. The Gentian violet is used as an inhibitor because it prevents the growth of most gram-positive bacteria. And then finish with this.
Hello, I'm Arad Lopez Huerta, and I'm going to talk about gram staining. Gram staining is a common technique used to differentiate two large groups of bacteria based on their different cell wall constituent. The gram stain distinguishes between gram-positives and gram-negatives groups by coloring the cells red or violet. Gram-positive bacteria stain violet due to the presence of thick layer of peptidoglycan in their cell walls, which retains the crystal violet these cells are staining with. Alternatively, gram-negative bacteria stain red, which is attributed to a thinner peptidoglycan wall which does not retain the crystal violet during the decolorization process. How does the gram staining work? Gram staining involves three processes: staining with a water-soluble dye called crystal violet, decolorization, and counterstaining, usually with safranin. Due to the difference in the technique of a peptidoglycan layer in the cell membrane between gram-positive and gram-negative bacteria, gram-positive bacteria with a thicker peptidoglycan layer retain crystal violet stain during the decolorization process, while gram-negative bacteria lose the crystal violet stain and are instead stained by the safranin in the final staining process. The process involves three steps. Cells are stained with crystal violet dye. Next, gram iodine solution is added from a complex between the crystal violet and iodine. This complex is a larger molecule than the original crystal violet stain, and iodine is insoluble in water. A decolorizer, such as ethyl alcohol or acetone, is added to the sample, which dehydrates the peptidoglycan layer, shrinking and thinning. The larger crystal violet iodine complex is not able to penetrate this tiny peptidoglycan layer and is thus trapped in the cell gram-positive bacteria. Conservatively, the outer membrane of gram-negative bacteria is degraded, and the thinner peptidoglycan layer of gram-negative cells is unable to retain the crystal violet iodine complex, and the color is lost. A counterstain, such as the weakly water-soluble safranin, is added to the sample, staining in red. Since the safranin is lighter than the crystal violet, it does not disrupt the purple coloration in gram-positive cells. However, the decolorization gram-negative cells are stained red. A gram stain is a type of microbiology or laboratory test that determines whether bacteria are present. It also determines whether bacteria are gram-negatives or gram-positives. The difference between gram-negatives and gram-positives bacteria can be important when determining appropriate treatment for an infection. Gram stains are performed on various types of specimens, including blood, tissues, stool, and sputum. Every student and professional who works with bacteria has to, at some point, learn how to do a gram stain. The gram stain is a stain procedure used not to only make bacterial cells more visible under the microscope, but also to differentiate cell types based on the cell wall composition.
Now, my name is Araceli Esparza, and I will talk about the general characteristics of the bacterium and how 6 animals of this bacteria, that is E. coli. E. coli, his germ is Escherichia. The general characteristics are that are short roots and gram-negative. The important species are E. coli, with the several serotypes, are distinguished based on the presence of the somatic antigens, capsular and flagellar. The habitat of this bacteria is in the intestinal tract of the animals and the man. The virulence are the syndrome enterotoxigen with the filling of the fimbriae. The production of the enterotoxins are ST and LT. The syndrome enterotoxemic is endotoxin LSP, the EDP, and the beginning of the disease edema. Another is cholecystemic syndrome, that is with the CLT, that is toxin lethal to the chickens. Then the plasmid cold B, the mechanism by the which confers increased virulence is not well clarified. The 6 in the animals are of this bacteria the enteric serotypes that are associated with the production of diarrhea and the pigs, cattle, sheep, goats, and horses. In the pigs, we are going to see about diarrhea newborn, diarrhea of the 3 weeks, and waning diarrhea, the edema, and disease. With the cows, we see the septicemia, endotoxemic, and the diarrhea. With the birds, we have the cholecystemia, cholegranuloma, and it is considered opportunistic pathogen in the urinary infections and respiratory tract mastitis, omphalitis, and other infections. Important culture, media, and biomedical tests. When we have this sick with some one of their animals, we need to take a diagnostic with these techniques. Like the rest of the enterobacteric, they are not demanding but a prepared use of selective and differential media such as macunkin agar, eosin, methylene, blue, and endo, among others. In the second, colonies have a characteristic green metallic sheen that distinguish it as fast fermentador lactose feature it shares with the two genders over Enterobacteria, that is, the Enterobacter or Klebsiella. Well, with this we are going to finish our presentation. That's all. Thank you for your attention.